Howdy people, thanks for checking in to MrUlrich'sLandofBiology.com. I am Mr. Ulrich. In this video, we're going to be taking a look at some of the ways that people have applied our knowledge of genetics and heredity to get things done. Now, this isn't a new thing. People have been using the principles of heredity for a long time, certainly long before we understood the molecular background to it. But now that we do know quite a bit more about how genes work and even how heredity works, we can certainly do some pretty neat stuff. People have known for a long time that traits are passed down from one generation to the next, and they've been using that knowledge for centuries. Through a process called selective breeding to domesticate animals, to get plants and animals to have the traits that they're looking for. And a pretty basic, pretty simple process, just going out there and saying, seeing which traits are the ones that are desired in the next generation, finding those individuals amongst the population that have those traits, and using those as breed stock for the next generation. What this guy here is doing is he's sexing the chicken. If you're going to breed chickens, you got to make sure that you have males and females, and it's not as easy from the outside to see. You have to take a pretty darn close look, so that's what that guy is doing. Now, if we're going to choose organisms that have the best traits, that kind of implies that all organisms are not the same. And of course, we're talking about all organisms not the same within a population or within a species. There are inheritable differences between organisms and individuals, and we call these inheritable differences variations. If you're thinking, wait a minute, selection, variation, and your brain is going ding, 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 wait a minute, did we talk about this in evolution? Yeah, there's a pretty striking similarity here between natural selection and selective breeding. In natural selection, it's the environment that's deciding or choosing which traits are beneficial, whereas in selective breeding, it's a breeder who is determining which traits are beneficial. Those traits might not be beneficial to the survival of the organism in the wild, and that's how we end up with domesticated pigs with ears that are so big that they hang over their eyes and they can't see. Traits like this certainly wouldn't persist in the wild, most likely. Now, we know that traits run in families, so if we are choosing individuals that are going to mate, that are going to contribute to the next generation, and we're choosing them based on traits that they have, well, since those traits tend to run in families, there's a good chance that those individuals are going to be related. And when we choose individuals that are closely related, we call that inbreeding. Now, a lot of people have a kind of visceral reaction to inbreeding. The first thing they think of are these gross abnormalities, physical abnormalities, oddities that come from inbreeding, and they often will even think of it as inbreeding as a mutagen, like it's going to cause some sort of mutation. And that's not the case. Inbreeding does not cause spontaneous changes in the DNA base pair sequence. What it does do, however, is it does increase the chances that we're going to have offspring that are homozygous recessive. See, if we choose individuals that are closely related, there's a good chance that those two individuals might both be heterozygous for a particular trait. And if those two individuals are heterozygous for a trait, there's a 25% chance that the offspring are going to be homozygous recessive. Now, in the case of blue eyes, something like that, not a problem. But quite often, these recessive traits, since they do come from the lack of a protein, sometimes that can be a problem. We like to think that our proteins all have functions. So if you're missing one of those proteins, we may have issues. So inbreeding, something to be avoided. It's probably why we have the trait as a population, as a human population, of thinking that it's gross. That kind of keeps us from having these, increasing the chance of homozygous recessive traits coming out in our populations. Now, that is not necessarily the norm for all other organisms. Inbreeding is relatively routine in out there in the plant and animal kingdom. The term hybrid, it's used in quite a few different ways. We can drive hybrid cars that has two different drivetrains, electric and gas. We can be hybrid for a particular trait that's going to have two different alleles for that trait. But in this case, we're talking about breeding. So what are we talking about as far as a hybrid here? Sometimes people recognize that the traits that they want in one organism happen to be found best in two separate species. So in some cases, very rarely, but in some cases, we can actually mate those two different species together. The cool thing that happens is quite often we get what's called hybrid vigor. You actually get an amplification of those traits that we were looking for. This case is really clearly shown in the mule. Right, a mule, like we talked about back in a unit on classification, a mule is a hybrid between a donkey and a horse. Somebody said, "Hey, I want the endurance of a horse and the strength of a donkey together in the same organism," and lo and behold, you end up with a mule, which is in some ways has more endurance and is stronger than either the horse or the donkey. Kind of neat. Now the drawback here, of course, is though they are close enough related to mate, they are not close enough related to mate and have babies that can make more babies that can make more babies. So we can put mules together, boy and girl mules together, Jacks and Jennies, play as much Barry White as we want. We're not going to end up with any baby mules. So up to this point, we've been talking about selecting which organisms are going to go ahead and mate and pass their genes on to the next generation. We also know that there's no guarantees when it comes to inheriting particular traits from parents. Now, with genetic engineering, using modern technology and our modern understanding of molecular genetics, we can actually get in there and manipulate individual genes and get them to do what we want. Several years ago, up in Scotland, at the University of Edinburgh, a team of researchers were able to clone a sheep using a transnuclear cloning technique. Now, what we mean by transnuclear is we're going to switch nuclei. So what they did is they took a body cell from the udder of one sheep, took the nucleus out of it, and threw the cell away. We don't need the cell. All we need is the nucleus. Remember, the nucleus of every one of your cells and every one of the sheep cells, except for the sex cells, are going to have a complete set of DNA, complete set of all the genes necessary for all of the structures in the body, all the proteins in the body. So we take that nucleus out of the body cell, took an egg cell from a completely different strain, different breed of sheep, took that egg cell and took the nucleus out of it and threw that away. Don't need that nucleus anymore. All we need is the cell. Took that nucleus from the body cell and stuffed it into the egg cell and gave it a little pulse of electricity, and sure enough, that egg cell with the new nucleus in it divided, formed a morla, and went on its merry way, and eventually we had a new sheep. And of course that sheep was genetically identical to the donor and had really no relation to the, other than its sheepness, no relation to the egg donor. And this is what we call reproductive cloning. Now, invariably, students will ask me, do we clone people? Well, sort of. Therapeutic cloning is used not just on people, but it is used with human cells. And this is how we keep germ lines of stem cells going, by taking those cells and growing them in tissue culture and using those cells to do research with. Even with cloning, we're still dealing with a whole genome. So we haven't quite gotten down to the gene level yet. Now we can. When we get into individual genetic engineering, we're going to talk a little bit more here about what we call recombinant DNA. So if we're going to take a gene from one organism and put it into another, that genetic material is going to be recombined with that genome, and we're going to refer to that as recombinant DNA. Back in the day, I don't know how many of you guys are old enough to remember this, but we used to listen to tapes all the time. And quite often, as you're driving down the road, listening to your fabulous new Tribe Called Quest album on cassette, you'd be rolling down the road, and of course the tape would get eaten. And you pull the tape out of the cassette player, and it's all crinkly and gross. And what we'd have to do is cut the crinkled section of tape out and tape the ends back together again so that we have one continuous piece of tape. And we can think of DNA a lot like cassette tape, right? It's linear. It remembers information in a long string, and different sections of the tape are going to be individual songs. We can even break it down to individual bars of songs. And back before people used cool and groovy software like Fruity Loops and GarageBand and some of these other things to make repeating loops of short pieces of music, laying cool beats over top of them and making music that way. We still could do that, we just didn't have the digital way to do it. We had to actually take a section of tape, find just that portion of tape that had the music that we wanted on it, cut that out, tape those ends together into another loop, and put that back on the tape machine, and that would play over and over and over again. And we called that was splicing. Now, because it's so similar to working with genes, we refer to it as gene splicing. When I was repairing my cassette tapes, I would usually use scissors or sometimes even a razor blade, but we can't use that on DNA. So what are we going to use? We're going to use what are called restriction endonucleases. These are special enzymes, and their job is to cleave the DNA, but they do it in a pretty special way. They do it not straight across the two chains. They're actually going to cut it in a way that's going to leave a few of those bases unpaired. And those unpaired bases, well, they really want to pair with their complements. And so that turns them into kind of sticky ends. And if we use the same restriction endonuclease on different pieces of DNA, well, that means that they're going to have the same sticky ends. And now we can start to rearrange and splice the ends of those DNA parts together. Now, these restriction enzymes are also used in gel electrophoresis, if you remember way back when we talked about this. Back in the beginning of the school year, we did our unit on tools of the biologist. We did talk about gel electrophoresis, so just to refresh your memories, gel electrophoresis is a way to separate and sort different sized particles of DNA. So how do we get the different sized particles? Well, we'll start out with a big long string of DNA, and we'll use a particular restriction enzyme to cut it into little pieces. And that will yield DNA of different sizes, little fragments that are different sizes. We take that combination of, that mixture of fragment sizes, and put them in the well, in one of the wells, turn on the power, and that's going to cause the particles of DNA to move through the gel. The larger molecules, because they are bigger and clunkier, don't go as far, and the smaller particles, which can move faster, actually go further through the gel. And so that starts to spread them out and sort them by size. Now, the way this is used most famously on the Maury Show to determine who's your daddy. The idea is, if two people are closely related, that means that those restriction sites, the order of bases that that particular restriction enzyme will cut at, those restriction sites should be in similar places if our DNA is similar. And that means that when we use the restriction enzyme, we're going to have similar sized fragments. And that would show up on the gel electrophoresis as similar banding patterns. Us humans, we're pretty crafty in the laboratory. We can do a lot of things. One of the things we can't do, however, very well is make proteins. Certainly make specific proteins. from scratch. So taking individual amino acids and gluing them together in just the right order to get the properly shaped protein that's going to do what we want to do, we're not very good at that. What we are really good at doing is getting other things to do our work for us. And that's what we're going to do here with genetic engineering with recombinant DNA. So we're trying to make a lot of a particular protein. What we need to do is find the gene for that protein and snip it out of the organism's genome. It can be an animal cell in this diagram here, but it can also be a plant cell or a fungus or another bacteria for that matter. We just need to find that gene, snip it out using those restriction endonucleases. All right. Then we're going to call on bacteria. Now, why bacteria? Because bacteria are really, really, really good at swapping DNA. And they do this by using little small loops of bacterial DNA called plasmids. So if we lerp out one of these plasmids and cut it using the same restriction endonuclease so that they have the right sticky ends, as we did with the target gene, it's really easy to get that target gene and that plasmid to what we call anneal, to stick together into one continuous loop of DNA, into one new plasmid. Now it's really easy to get that plasmid back into a bacterial cell. And now we can clone that bacterial cell, or we don't even have to clone it, we can culture that bacterial cell. Since they're asexual, they're going to go through simple cell division, and all of the bacteria will tend to have this little loop of DNA that contains the gene for the protein that we're looking for. And of course, bacteria are stupid by definition. They can't write poetry or music or think about the future or reflect on the past. All right, they're just dumb bacteria. They're gonna do transcribe and translate all the DNA that's in there, whether it's their own or this recombinant DNA, and they're gonna go ahead and synthesize the proteins. One of the applications of this recombinant DNA technology is making farm products like BT corn. Now, this is a type of corn. It's been genetically modified. The BT stands for Bacillus thuringiensis. Bacillus thuringiensis is a pretty common soil bacteria that happens to have a gene that codes for a protein that is toxic to insects. In fact, it makes the insects' digestive tracts pretty much fall apart, and then they die. So what they figured out to do was, using restriction enzymes, cut the code, the gene for this BT protein out. They used the same restriction enzyme to cut into corn genome, corn DNA, and with the same sticky ends in place, are able to pretty easily stuff that BT gene into the corn genome. And then from a cell that has this, a single corn cell that has this BT gene in it, they then clone a corn plant and then asexually grow countless corn plants from the one modified corn plant. And that way, every single cell in that corn plant is going to have a copy of this gene in it, and is going to produce this protein that, when insects eat it, causes them to die. This has some pretty obvious benefits. If the plant makes its own pesticide, then we don't have to apply pesticides. The plant makes its own pesticide inside of the plant, so quite often most of the pesticides that are applied don't actually end up on the plant. They end up in the soil and end up in the air. And so this way we don't have to apply them, and the environment benefits from not having the insult of pesticides poured on it all the time. Makes things cheaper for the farmer to produce, and therefore cheaper for people to buy, and everybody's happy, right? Well, not everybody's happy. The European Union has actually banned genetically modified crops like BT corn. They have some concerns, both health concerns for people who eat them, although that hasn't been conclusively shown, to my knowledge, in research. There's research on kind of going both ways there. What we have figured out, though, is that the BT protein doesn't discern between pests and pollinators. So what ends up happening is pollinators that we like, those insects that are going to carry pollen from one plant to another and responsible for making about 2/3 of the crops that we have, they're also dying too. So there's definitely a cost-benefit analysis that needs to be done. I'm going to leave that up to you to do. So educate yourself, look into the issue, and you make your own decision. We don't always have to use bacteria to do our dirty work. We can use other organisms too. Now, some work of some researchers up in Plattsburgh, New York, led to the development of a product called BioSteel. What these researchers recognize is that spider silk, famous for its strength and lightweight properties, would make an excellent fiber for textiles. But that would also mean they would have to grow a lot of spiders, and spiders can be a little oogie to some people, so there's got to be a way around it. So what they did, of course, was they took the gene that coded for spider silk and they stuffed it into a goat nucleus. Took that goat nucleus and put it into an egg cell and cloned on its way and made an actual goat that contained the gene for spider silk. Now, it's not like these goats spun webs and swung around the city fighting crime. Spider goat does whatever a spider goat does. No, instead, they secreted the milk. They secreted in their milk the protein for the silk. And so all we had to do was get the protein out of the milk, purify it, and then use it now in applications like Kevlar, in bulletproof vests, and other things like that, where we really need strong stuff that's flexible. We don't always have to use bacteria to do our dirty work. We can use other organisms too. Now, some work of some researchers up in Plattsburgh, New York, led to the development of a product called BioSteel. What these researchers recognize is that spider silk, famous for its strength and lightweight properties, would make an excellent fiber for textiles. But that would also mean they would have to grow a lot of spiders, and spiders can be a little oogie to some people, so there's got to be a way around it. So what they did, of course, was they took the gene that coded for spider silk and they stuffed it into a goat nucleus. Took that goat nucleus and put it into an egg cell and cloned on its way and made an actual goat that contained the gene for spider silk. Now, this time we're going to go back to using the bacteria, this time E. coli, and it's basically the same process that we went through with BioSteel and that we introduced this topic with. You get that human insulin gene, we could turn it into a plasmid, and since those plasmids are really easy to get into bacteria, all we have to do now is culture those bacterial cells with those plasmids and lyse them, and we kill them, basically, kill a bunch of them, break them open, and separate the insulin from the culture, and poof, we have human insulin. And like it says, they're happier camels. With the Regents exam right around the corner, it's probably worth our while to take a look at an example of a Regents question. We might want to hit pause here. Hopefully, you recognize that we're talking about bacterial cells that are able to synthesize insulin. Well, that about wraps it up for our unit on genetics. Thanks again for stopping in to Mr. Ulrich's Land of Biology.com. Once again, if you have any feedback, please feel free to email me. Go to my website, Mr. Ulrich's Land of Biology.com, to get the email address, send me the feedback, and to download other supporting material, vocab lists, notes sheets, other handouts and diagrams. It's all on there. So thanks again for checking in, and we'll see you in class.